Linknya Kak kasih ya. Kak ini.
Good morning, Dr. Heng. Welcome to the webinar.
Hi, good morning.
Good morning.
Good afternoon for you, huh? Hi, Vishnu.
Oh, yes. Good afternoon. Yes. Today, Mr. Musofan, who will become the moderator for this webinar, Dr. Heng.
Yeah.
Good morning, Dr. Heng.
Good morning.
How
There
are you?
Yeah. I'm good. And how about yourself?
I am good.
So you are...
There is 2 more minutes. So we will wait 2 more minutes, Dr. Heng, before I
That's
start
okay.
the guest lecture.
Yeah. So you are hosting the meeting, right?
Yes.
Okay. Yeah.
Okay. Good afternoon, ladies and gentlemen, and good morning for Dr. Heng in Netherlands. We are delighted to welcome all of participants to the International Guest Lecture 3-in-1 program entitled Understanding Seed Physiology, Development, Germination, and Dormancy. This guest lecture is aimed to gain a better insight into seed physiology. It is an honor for me to introduce and welcome our distinguished key speakers, Dr. Henk Hilhorst from Wageningen University, Netherlands, and Mr. Danang Widyarso, SPMSc from PTBC International Tbk, Indonesia. Thank you for the time-giving guest lecture to our department. And also welcome all participants, students of Agronomy Department, Brawijaya University. Ladies and gentlemen, I would like to inform the agenda of today's guest lecture. The first one is listening to national anthem of Indonesia. Second one is opening remarks from the head of department, Dr. Nurrahmi Ardiarini, SPMSI. And next is presentation from Dr. Henk Hilhorst, and then followed by question and answer. The next agenda is second presentation from Mr. Danang Widyarso, SPMSc, and then followed by question and answer. And the last agenda will be closing. Ladies and gentlemen, now we are in the first agenda, listening to Indonesia National Anthem. Please be seated upright and listen I'm sorry, there's a technical problem. We will replay again for the national anthem. Okay, ladies and gentlemen, next agenda is opening remarks from the head of department. I would like to invite Dr. Nurrahmi Ardiarini. Time is yours.
Thank you, Mr. Musofan. Assalamualaikum warahmatullahi wabarakatuh. Head of Plant Physiology Laboratory, distinguished guest speaker, ladies and gentlemen, good morning for Dr. Health. Good afternoon. It is with great pleasure that I welcome you to this online guest lecture held by the Department of Agronomy, Faculty of Agriculture, Brawijaya University. Let me begin welcoming for our guest lecturer, Dr. Henk Hilhorst from Wageningen University and Mr. Danang Widiarso MSc from DC International TBK, who will address the guest lecture 3-in-1, a program with their unique perspective on the topic. I also would like to thank our guest lecture committee who have been tirelessly coordinating and organizing the event in spite of their own tight schedule of lecturing online classes, all for the success of the guest lecture, which I believe will provide a venue for all participants to gain insight from one another and to explore new opportunities for further collaboration. During this guest lecture, we will discuss the understanding seed physiology development, germination, and dormancy. The key challenges to tackle here include the advancement of technology such as development, germination, and dormancy on physiology seed. Can physiology seed meet the new challenges of the new century with a new understanding? And if so, then how? Or if not, what will do? So ladies and gentlemen, today speaker will address issues such as understanding seed physiology inclusivity, response, and institutional collaboration in agriculture. What we need is not just to conclude that this is important, but to find meaningful tools and to make things happen. In this online guest lecture, we represent educational institution, academy, practitioner, and student. My hope is that this day give as an opportunity to build stronger ties between academia, multilateral sector, and the agriculture service in Indonesia and beyond. So we all can contribute to improve agronomic and education for all in the coming years. Thank you for me. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Nur Rahmi. Now we are coming to our main agenda, presentation from keynote speakers. Yet, before beginning the presentation, I would like to inform a brief information about Dr. Hang Hil Horst. Dr. Hang Hil Horst is Associate Professor in Plant Physiology. He is a member of Department of Plant Sciences, Laboratory of Plant Physiology, Wageningen University. His expertise are molecular genetics, plant molecular biology, plant physiology, and seeds. He has published extensive research articles on seeds germination and performance. Today's guest lecture is divided into two parts. The first part is presentation, and the second part is question and answers. Dr. Hang delivers his presentation in 40 minutes, and then question and answer will be around 20 minutes. Questions can be addressed through chat or be asked directly by raise hand. So participant can ask question through chat or comment in YouTube, or can ask directly by raise hand first. Without any further ado, I would like to invite Dr. Hang Hil Horst. Time is yours.
Okay, thank you so much for your introduction, and also I want to thank you for inviting me to be a guest speaker today on this this webinar. And I will start my lecture by sharing the screen. And I have to do this. Okay, I assume that this is fine for everyone.
Yes. Yes, Dr. Hang.
Okay, then I will start. So when I was invited to speak about seeds, and I like that always because I am a seed scientist, of course. The question, well, I was asked to talk about development, germination, and dormancy. And that is a lot.
Dr. Hang. I'm sorry, Dr. Hang, could you please change the pointer into laser?
The pointer, yes. Okay, like this? Yeah. So
yes,
I was asked to give a lecture about seed development, germination, and dormancy. And that, of course, is a very broad field. And I would need, I don't know, maybe 20 lectures to cover all that. And because basically this is also the title of a book I have been co-authoring 2013. And so instead of trying to do everything, I decided to talk about seed physiology in relation to seed quality, because you are a department of agronomy, and I think that seed quality is of eminent importance to agronomy in general. So I hope you like it, and let me start. So to begin with, seeds are the basis of agriculture. We all know that. And because of that, also of modern civilization. Because our modern civilization could only begin when people discovered that seeds could be stored and could also be transported. And of course, the seeds are important as food, like cereals and beans, they're all seeds, but also just as importantly for propagation, to start a new crop. And then sometimes we tend to forget this, but also this is becoming very important nowadays in relation to climate change and so on. Seeds are the most important conservators of biodiversity. And that is because you can store them for a very long time under dry conditions. And you may have heard of the Svalbard Global Seed Vault. It's up in the Arctic Circle, where seeds are stored, well, hopefully for thousands of years to come. Now, as you can see here in this list, the most planted crops in the world, the first 6 or 7 are all seed crops. This is the seeds that we use from these crops. And all the others are non-seed crops. But this tells you already how important seeds are. Of course, wheat, maize, and rice are the most important ones in the world. Most people in the world actually depend on these crops for their diets, their daily diets. So I want to talk about seed quality in relation to seed science. What is seed quality? That is an important thing to keep in mind. Seed quality for a company, a seed company, or for a farmer relates to the performance of the seed in the field, but also in storage. Also, farmers want to store seeds the next year, resulting in a successful crop under varying conditions. And varying conditions mean also unfavorable conditions. So that is quite a challenge to reach high seed quality. And for that, a lot of research is being done. It is applied research, but just as much fundamental research. Because seed quality traits refer to several biologically important processes, like seed development, germination, growth of seedling, seed aging, etc. And that also means that seed quality is not a single trait, but a very complex trait. So seed quality cannot be improved by breeding for one gene. It is a whole bunch, a whole complex of genes that is underlying seed quality. Now here are a couple of examples of seed traits that are involved in the total picture of seed quality. So the biological process growth or arrest of growth is referring to germination or the germination potential of seeds. So germination under optimal conditions, and most importantly, germination under stress conditions, which is also called vigor. So seeds with high vigor are able to germinate and to produce seedlings under stress conditions like drought or salt. And then dormancy. And dormancy, of course, is an arrest of growth. Then there is the phenomenon of seed aging. Seeds can be stored for a very long time, and we come back to that later on. But also seeds in the dry state, they age, and in the end they will die, like all living organisms. But to be able to stay alive for such a long time, they have to withstand to be dry. And to be dry refers to a phenomenon that we call desiccation tolerance, the ability of seeds to be dried completely. And then the relation with embryogenesis, so the formation of the embryo in the seed, also growth and development. For instance, seedling morphology is extremely important in agronomy. Seedlings have to be normal, shoot size and root size are important. The root growth rate, of course, all related to vigor, root architecture, and maybe the most important quality criterion for seed companies is the usable transplants that are generated by the seeds. And then, of course, reserve food in seeds, proteins, lipids, sugars, relate to seed size and seed mass, and that also relates back to vigor. And of course, the reserve food also represents the nutritional value of many crops. So seed quality is a polygenic trait. So many genes are involved to establish seed quality, or seed performance as we prefer to call it, and seedling establishment. So there is genetics. So you have cultivars that have good germination, cultivars that have bad germination, no dormancy or dormancy. The environment is important. Stress conditions are important. The harvest, the timing and the method of harvest. Also post-harvest treatments. Also storage conditions. Also epigenetics in relation to genetics. They all contribute to the final performance of the seed and the establishment of a seedling. So let's have a look at the life cycle of plants. Here you have the life cycle, and of course starting with fertilization, and from there the seed starts to develop after successful fertilization. And then the seeds become desiccation tolerant. They can be dried. The seeds may become dormant. I will come back to that later. And then we finally have a ripe seed or a mature seed. And that seed can be stored under dry conditions for a long time. And then at a certain moment, a farmer will decide that the seeds will have to be sown and it will have to germinate. So these are all the different processes in the life cycle of plants related to seeds. And you can see that is the bigger half of the whole cycle. And then, of course, as mentioned, the environment plays a role and influences all these different processes. Again, underlining how complex seed quality is. Here you see a picture of seed development. This seed is from Arabidopsis thaliana, which is a model species for plant scientists. But basically, what you see here is something that you find in all the dicotyledonous plants. The DAP stands for days after pollination, and you see that this species has a very quick seed development. It starts with a very small embryo here, a pre-globular embryo. It grows to a globular embryo, then it becomes a heart-shaped embryo because you can see the beginning of the two cotyledons. Here is the embryo, a bit larger, and you can see that the endosperm solidifies. And then here you have the embryo again. It is growing at the expense of the endosperm. The endosperm is completely digested until you have an embryo that is occupying most of the seed. Well, as I said, this is for Arabidopsis thaliana, but this building plan for seeds you will find in all dicot species. For monocotyledonous species like cereals, of course, it is different, but it is comparable. The different stages are comparable, also at the molecular level. And the seed development is subdivided always in the morphogenesis or embryogenesis, the establishment of the embryo body plan. And from here, the process called maturation starts. During the maturation phase, the embryo only grows and there are no more cell divisions. So the cell differentiation is completed here. The embryo is actually only swelling until the seed is mature. And during the maturation phase, a lot of important things happen. There is an accumulation of storage proteins and also of lipids. Photosynthesis is stopping. There is an acquisition of desiccation tolerance and also a preparation for dormancy. And basically here, in the maturation period of the seed quality is acquired. And you can see that here, you look at seed development. Here again, you have the different phases, but you can follow seed development by looking at the seed weight. And of course, the seed is growing bigger, and that means it's becoming heavier. So here, the seed is still small, and it's increasing in weight. But then when there is reserve deposition, of course, the weight increases quite a bit. This is the fresh weight. Now, if you look at the dry weight, of course, it's also increasing. This is the proteins and the lipids that are accumulating in the seeds. And at the same time, you can see that the water content is decreasing. And that is because proteins and the starch and the lipids, they occupy cell space where there can be no water. So the water is driven out of the cells. And then finally, in many species, there is a further drying stage until very low water content. Here you see the same picture again, but during maturation, functions are acquired that determine the post-harvest seed phenotype, which is called seed quality. And that's what you can see here. Very early during development, seeds become germinable. The moment the cell differentiation in the embryo is completed, the embryo can germinate, but it needs to be germinated on a medium. It needs nutrients. Later on, the reserve food is, of course, accumulating, and then germination of the seed can occur without nutrition from the outside. Also, a bit later, seeds become desiccation tolerant, so they can be dried. Later, the vigor of the seeds is acquired. So this is the major seed quality trait that is developed here. And then usually at the very last, the storage life, the capability of seeds increases. Seed quality is influenced by the environment. I mentioned it already. And that is true for the vegetative phase of the crop and also during grain filling. So the seed quality is influenced by the environment when the seeds are developing on the mother plant. But also, of course, when the seed is dispersed. So the environment has a double effect. During seed development on the mother plant, and then after the seeds have been sown in the field, of course, then also it really matters if there's water enough, if there is light, if the temperature is right. Well, here you see some factors of the environment that influence seed quality. Temperature is one of the most important ones, and we come back to that one later. The photoperiod and like day length is important. Light quality is important. It really depends on whether it is cloudy or not, whether seeds are germinating under a canopy, under other vegetation or not. Water stress is important. How much water is available? Nutrients from of course, from the soil, potassium, nitrogen, phosphate. But also the position of seed in the fruit, and the position of the fruit on the plant, the age of the mother plant, and so on. And again, this influence is occurring during seed development, but also when the seed is sown in the field. Now, there's another problem, and when you look at seeds, and that is that the natural and the derived functions of seeds are conflicting. So natural seeds, seeds that of wild species that you find in the field, when you put them to germinate under optimal conditions, you can see very often something like this. This is the germination time curve, so germination percentage against time. And a natural seed germinates slowly, and not all the seeds germinate. And so part of this seed population is dormant. And this is normal, and this is because under natural conditions, seeds spread the risk of premature death over time and space. So you can imagine if all the seeds dropping from a plant would germinate all at the same time, and then a disaster happens like a hailstorm, the whole population will be wiped out. In this case, it is only part of the population that will die, and the rest will stay dormant and will wait for another moment to germinate. So this is the natural condition. But if you look at an ideal crop seed that you sow in the field, you want it to germinate very quickly. You want them to germinate for 100%, and you want them to germinate very uniformly. So you will end up with a very uniform crop in the field. And this is conflicting. So the seed industry, but also farmers, what they try is to get from this situation to that situation. So the question is, how can we dissect this complex trait into its components, study these, and use these results of fundamental research to improve seed and, of course, also crop quality. So the development of seed quality during seed maturation depends on the accumulation of food reserves, as mentioned, for vigorous seedling growth, the acquisition of dormancy, and that is related to the speed of germination and the amount of germination, and also on the acquisition of desiccation tolerance for long-term dry storage. So we will start talking a little bit about dormancy. When you talk about dormancy, you have to talk about a plant hormone, which is called abscisic acid, or ABA abbreviation, and also gibberellins. And these two play a major role during seed development. So here you see an ABA molecule. An ABA is a plant hormone that inhibits growth. On the other hand, there is the gibberellin, GA, that promotes growth. And those two are so-called antagonists. So this one inhibits growth, this one promotes growth. And if you look at seed development, you can see that ABA and GA are associated with a number of seed developmental events that are important for seed germination. So here you see the amount of ABA during seed development. You see here are two peaks, and at the end of development, ABA levels are very low. GA levels are high in the beginning of seed development. They are low throughout development, and then they become high at the end of seed development. And you can see that these hormone levels are related to the synthesis of seed reserves, desiccation tolerance, induction of dormancy, and germination. So germination is growth and is related to gibberellins, because here you have more gibberellins than ABA. But here, these processes are related to high levels of ABA. So there is a good chance that ABA is regulating these processes. Also, and I come back to that, a phenomenon called viviparity, that when seeds germinate before they are mature. Here a very general picture of the ABA content and germination during development of tomato seeds in this case, but this could also go for many other species. So here you have the germination percentage, the days after pollination, seed development, and here the ABA contents. Excuse me. So as I said before, seeds become germinable quite early in seed development. Take them out of the fruit, they will germinate for 100% at this point. But later on, the germinability decreases until a point here where no seed will germinate when you take it from the fruit. So these seeds are dormant. If you look at the ABA content, you can see that when the ABA content increases in the seed, the germinability decreases. Here the ABA content is very low, and then the seeds lose their dormancy gradually. This is also a very general picture that you find in many, many species. Now, if there is no ABA or if there is a lack of sensitivity of the plant to this hormone, you may see viviparous germination. And another name for viviparous germination is pre-harvest sprouting. I will come back to pre-harvest sprouting. It's very important for agronomy. As you can see here, these maize seeds, corn grains, are germinating on the cob. And of course, that is very damaging to your harvest. You cannot use these seeds anymore for any purpose, for baking or whatsoever. Here in tomato, you also see that the seeds have germinated inside the fruit. This may happen in a very overripe fruit, but also a fruit from, for instance, a mutant of tomato, which is not able to make ABA. And then you can see it germinates inside the fruit. Viviparous germination or premature germination. So what may appear from what I have told you is that the ABA-GA balance is decisive for germination to occur, both during development but also following development. So we literally see it as a balance. ABA related to seed development and dormancy, GA related to growth, seed germination, and the absence of dormancy. And this balance determines whether seeds will stay dormant or if they will germinate. So how can you break dormancy? There's a very simple seed treatment, very effective and sometimes necessary when you want to sow your seeds. Sometimes it is just enough to store the seeds dry for a while. We call that after ripening, and that may take a long time. It may be weeks or months or sometimes even a year. But during this after ripening, the seeds lose their dormancy, so they go from this germination curve to that one. But also a low temperature treatment of the hydrated seeds can be effective. We call that cold stratification. Usually that takes a couple of days. And then you go from that curve to the ideal curve, germination curve. The effects of the environment on seed germinability have been described for the first time in a more systematic way around 30 years ago by Michael Fenner from the United Kingdom, where he says that the growing conditions of a parent plant may affect the degree of dormancy of its seeds. And also lower dormancy, so increased germinability, is generally associated with environmental conditions during seed development like high temperatures, short days, red light, drought, and high nitrogen levels. So under these conditions, seeds have a low dormancy. Now I have one example here that shows it very well. Again, this is Arabidopsis thaliana, not a crop, but a very useful model species. This is the plant, it's a wild species. What you see here is the germination frequency, germination percentage of seeds that were grown, well, from plants that were grown at 20 degrees, plants that were grown at 15 degrees, and plants that were grown at 10 degrees Celsius. And here you see the number of days to break dormancy. If you look at the seeds that were developed at 20 degrees, you see there's no cold stratification required. There's 100% germination. Those seeds are non-dormant. Dormancy is totally absent. When you grow the plants at 15 degrees, you see you need something like 10 days, 12 days of cold stratification to break dormancy. So this dormancy is deeper than that dormancy. You grow the plants at 10 degrees, the dormancy is even deeper. And you see that even 2 weeks of cold stratification are not enough to break dormancy. So that is interesting. That is a very good example of how the maternal temperature influences the dormancy level of the seeds. Now, if we look at the amounts of ABA and GA in the seeds, then you can see at 22 degrees, well, that is close to this one, but there is no dormancy. There is relatively low ABA and high GA levels. Sorry, ABA is low at 22 degrees, but it is high at 15 degrees. 15 degrees is more dormant than at 22 degrees. The GA levels are just the opposite. At 22 degrees, they are high as compared to 15 degrees. So this is a very clear example of this ABA-GA balance. So at 22 degrees, the balance is on the GA side. At 15 degrees, it is on the ABA side. Yeah, so a very clear example, almost quantitative, of this balance. Now, if there is too little dormancy, there's also a seed quality problem. And in that case, I'm referring to precocious germination of cereal grains prior to the harvest. It may happen when conditions are cool and moist and close to the time of harvest. This causes economic losses in grain quality for baking, and it is particularly prevalent in cultivars with low grain dormancy. So there is a very clear genetic component here. White wheat has less dormancy than red wheat, so there is a genetic component. Here you see some examples of pre-harvest sprouting. This is a big problem in many countries of the world. So there's a lot of research going on to find out the causes of pre-harvest sprouting. And well, one of the main conclusions is that it is a matter of low ABA or low sensitivity to ABA during seed development. And it is very likely that this has occurred during domestication and breeding of the crop, because there has always been a selection for quick germination. And we know that quick germination is related to low ABA. If you do that selection again and again and again, you will end up with seeds with very low ABA. Yes, they germinate very quickly, but they may even germinate during seed development. And that is an unwanted situation. So there's a lot of research going on, as I said, particularly using quantitative genetics to find pre-harvest sprouting loci. And there's a lot of fine mapping going on at the moment in different crops, in wheat and in barley. And it seems that in a few years or so, because using this map-based cloning, the exact loci or genes may be found, and then can be used in breeding programs to breed out the pre-harvest sprouting. Now, a very recent, a very promising development to resist pre-harvest sprouting can be reached by genome editing. You may have heard of a technology called CRISPR-Cas9, where you can edit the genome in a very, very precise way. Now here you see a real nice example in wheat, hexaploid wheat, where using genome editing in the DNA, these three Gs on these three genes were replaced by an A, a T, and a T. So single base mutations introduced by using genome editing. And then you can see that here there's no pre-harvest sprouting, while here at the same developmental stage, there's a lot of pre-harvest sprouting. So actually what you do is you reintroduce dormancy. I think this is very spectacular. It's quite recent, it's 2019. So that is very promising, and it's a lot quicker than using quantitative genetics and breeding and conventional breeding. Okay, I have to move on. Acquisition of desiccation tolerance and longevity for long-term dry storage, also very important. Seeds are an important survival strategy of plants. The mature dry seeds can withstand extremes of drought and cold because of their low water content, whereas the parent plant will suffer frost or drought damage and even then maybe even fatally. How long can seeds survive in the dry states? Well, very long, maybe tens of thousands of years, but really proven experiments show that in this case, lotus seeds may survive for about 1300 years. And the seed germinated, but the seedlings were quite poor. And this example is even more extreme. This is the date seed, the Judean date palm of Masada is the oldest ever to sprout. And this seed was determined to be 2000 years old. And of the seeds that were found, one produced a viable seedling and produced, you see, a very healthy, viable palm tree. So seeds can be stored at least for hundreds of years. And therefore we have gene banks in the world, around 1700 in total. The most famous one is the Svalbard gene bank in Norway, north of the Arctic Circle. This is the global seed vault, has been in the news for a long time. Here you see the inside of a seed bank where seeds are stored under very controlled conditions, low relative humidity, low temperature. And here, this is a seed storage facility in Fort Collins in the United States where seeds are stored in liquid nitrogen at very low temperatures. And under such conditions, the seeds are not notably aging at all. So they can be stored indefinitely. In orthodox seeds, drying is integrated in their normal development, and so the seeds become desiccation tolerant. And that is necessary because of this drying phase in many seeds. They have to be tolerant to survive that. And you can see how that develops in barley in this case. In this experiment, embryos were excised from the seeds and put to germinate. And here you can see at very early stage, germinability increases. But these are fresh seeds. But if you dry the seeds first and then put them to germinate, then what you see is also an increase in germinability, but a week later. And that means that in this species, desiccation tolerance is acquired in this week, well, these 5, 6, 7 days of seed development, because here they have become fully tolerant. There's a nice example. Medicago truncatula is it's a legume. You see here seed development 12 to 44 days after pollination. You see the water content is dropping and the dry weight is increasing, as I have shown you before in a different graph. And here the desiccation tolerance was measured. And you see it is increasing here. And it is 100% maximum at the moment that the water content is becoming very low. Quite remarkably, you can see that the storability of the seeds or the longevity of the seeds is increasing much later. So seeds have to become desiccation tolerant first, then you can store them, but also the storage ability, the storage capacity is increasing, but much later during seed development, during maturation. This is desiccation tolerance and that is longevity. Some seeds don't survive drying. They have to stay in a hydrated state. And some of these are commercially very important seeds, like citrus seeds or coffee seeds or cocoa seeds, wild rice as well. So those seeds, and we call them recalcitrant seeds, cannot be stored dry, cannot be stored in seed banks. And that is a big problem. Particularly now with our loss of biodiversity, we want to preserve germplasm, and it is very difficult with these recalcitrant seeds. The seeds that can be dried, the orthodox seeds, they have mechanisms to protect themselves from damage during drying. particularly because cellular membranes and macromolecules like DNA and RNA and also some proteins are very sensitive targets of desiccation damage. Desiccation damage in a dry state is usually caused by oxidation, by free radicals, by reactive oxygen species. Some protective compounds, I will not go into the details, that you find in seeds are sucrose and oligosaccharides, is a different kind of sugar. Lots of stress proteins like ureas and heat shock proteins, and also, very importantly, antioxidants. Now, I want to end my lecture with a few words on climate change and agriculture. And you have, I'm sure all of you have seen these maps, the projected impact of climate change on agricultural
Dr.
yields. Yes?
Dr. Heng, I'm sorry, your time is 5 more minutes. 5 more minutes. Thank you.
I'm almost done. Okay? Yeah. And as you can see, your country is also in danger, and the projected decrease in yield is something around, well, let's say 10%, which is a lot for major crops like rice and wheat. So how does seed performance change in a changing climate? And if it is changing, what are the consequences? And that is, I think, a focus of many seed biologists nowadays, to see what the consequences are of climate change and what can we do about it to preserve seed quality and crop quality and yield. So for the wild seeds, is biodiversity at stake? So will we lose species? And for crop seeds, is food security at stake? That is, of course, a very important question. And as you have seen in the map, yeah, food security may be at stake when yields are going down too much. So the greatest challenge for the future, also for seed biologists and plant physiologists, agronomists, plant breeders, we have to design new crops, but also new seeds to produce these crops that are adapted to climate change. And you may call that climate smart agriculture. So what you would like to do is to have plants, crops that are able to grow under very harsh conditions. Well, that is a huge challenge. For that you need plant breeding, of course, like we have been doing for thousands of years. Fundamental research, particularly integrated molecular genetics and genomics. And with that as a basis, we can use plant breeding and CRISPR-Cas, genome editing, to improve the crops. But also to improve the quality of seeds, we can use biologicals and chemical stimulants. So not improving seed quality by genetic means, but by other means. I will have 1 or 2 slides about that. So design new crops, as I have said. The basis is the integrated molecular genetics and genomics, so fundamental science for genetic improvement by either plant breeding, which is a slow process, or by genome editing, for instance, by CRISPR-Cas9. And that is a quick process and a more precise process. But of course, if you want to use this gene and genome editing methodology, you need to know which genes you will have to edit. And for that, you need this fundamental science. Finally, you can also improve seed performance by seed treatments. Conventional means, you may have heard of it, seeds can be primed. Seed companies do that a lot. Seeds may be treated by different temperature treatments, or seeds may be coated. So you add pesticides or growth regulators to the seed for better performance in the field. But nowadays, a novel approach is to use biologicals. And that is where the future of applied seed science will be for the next 10 years or so. What are biologicals? They are naturally occurring organisms plant extracts, or organic substances that complement the seed's genetics and traits to enhance plant vigor and establishment. So everything that is natural, organisms like bacteria or fungi, plant extracts, or organic substances. And the results are sometimes spectacular. They may increase nutrient availability, so the take-up of nutrients from the soil, development of the root system of the crop, suppresses diseases and pests, and also maximizes the genetic potential of the seed. So it really employs potential of the seed. And it also promotes sustainability because most of these biologicals are environmentally friendly. So this is a huge future for seed technology. And at the moment, in many countries, people are screening hundreds and thousands of these biologicals for their performance in different crop seeds. And these biologicals are added to a seed coating. And there are different types of coating, but here you see an example, a cross-section of a seed with with the different layers of coating. So it can be insecticides, it can be a fungicide, rhizobia, this is for instance an inoculant with bacteria, can also be taken up in this coating, growth stimulants, nutrients. So the whole seed is packed in all kinds of things that are good for the seed and for its performance. And then you can also give it very nice color to distinguish the different species, different lines and cultivars, but also for seed companies to have their own marker, their own color. Even golden seed, as you can see. And I think that this was my last slide, so pretty much in time. And I want to thank you for your attention.
Okay, thank you, Dr. Hang, for a comprehensive presentation. Now it is time for question and answer session. Question can be addressed through chat or comment if you are in YouTube, and also asked directly by raise hand. And now we have one question from Dr. Sutini, UPM Veteran Jatim. The question is, please describe to overcome dormancy in Camellia sinensis seed.
Okay, thank you for your question. I don't know Camellia sinensis seeds. Are these probably seeds with a very hard seed coat? I really need more information to say something about it. I don't know these seeds, so I don't know what treatments could be beneficial. Maybe Dr. Sutini can help me with that.
Okay, Dr. Sutini, probably you can give some clue about the one. Camellia sinensis. Camellia is a kind of tea.
Yeah, but I don't know what type of seed it is, so I can say no. I can't say much about how to break its dormancy. So I need more information about how what the seed looks like.
Okay. Thank you for your response, Dr. Hang. While waiting for another clue from Dr. Sutini, I will invite another questions from other participants. You can ask directly by raise hand, or you can address the question through chat.
Well, people are still digesting the lecture, I think.
Yes, or try to typing, still typing.
Hello, excuse me, can I have a question?
Sure.
Okay, yes, sure.
Okay, my question is that...
Your name first, please.
Okay, my name is Genesiska from Special Region of Yogyakarta, UMY. Sorry. how we can indicate that our seed need have to decrease the abscisic acid. I mean, if we have the seed, then we storage in the place, and then we will germinate. But this fail. How we indicate we can give, for example, for the gibberellic acid.
Yeah, that is a very good question.
Yes,
But what you will see is when you store seeds dry, they will lose their dormancy slowly. And if you would measure ABA contents, you can see that the ABA contents decrease. But in many cases, yeah, people ask a lot, well, if the seeds are dormant, you can just give them gibberellins and then they will germinate. And in some cases that is true. But sometimes when seeds are dormant, they are not very sensitive to gibberellins. So then you can give them gibberellins, but then still germination will not be very good. Okay, another disadvantage of giving seeds gibberellins is that once the seeds germinate and the gibberellins are still there, the seedling will grow too fast, so you get very long but very thin seedlings that are not very vigorous. Yeah, so my answer is sometimes you can give GA to seeds and they will germinate better, but in many cases it doesn't work.
Yeah, that's happened to our seed, that it doesn't work for giving the hormone of gibberellin. So is there any treatments for that? In here is maize seed.
Sorry again?
In this case is for maize seed. Maize
seeds.
corn. Yeah.
Oh, that is strange because maize seeds are never dormant. In maize seeds, the dormancy has been selected out for a long time already. So maybe there's another cause of bad germination.
Okay. Okay, thank you.
Yeah, you're welcome.
Okay, thank you, Bu Janesiska. Now another question is from YouTube. The question from Jizul. May I ask how to prevent seed dormancy?
Sorry again. May I ask how to
prevent seed dormancy?
Oh, how to prevent dormancy? That is also a very good question, and that is a question that a lot of people do research in. As I have told, dormancy is very much influenced by the environment, the maternal environment. And so it's very unpredictable. So one year your seed harvest may be non-dormant, and the next year it may have quite a bit of dormancy. It is, of course, most cases it is not possible to change the environment. So there is now research going on to make the plants, the seeds, less sensitive to the environment. So if the seeds develop independently from the environment, that would solve your problem. But that will be still a lot of years of research before we get there. But that is one approach that is taken. Otherwise, there's not much that you can do about. If you cannot change the environment, you cannot change the dormancy, unless you start a breeding program to breed out dormancy, like has been done for most crops. Actually, they don't have dormancy anymore. So by genetic means, but that's also a long program.
Okay, thank you for the response, Dr. Hang. Now we have Mr. Arifin. Raise hand, Mr. Arifin, you can ask the question. Mr. Arifin. Okay, let's go to another question from Richard Tran. If the world was post-apocalyptic, what seeds would you seek out the most?
Sorry, again, what is the question?
If the world was post-apocalyptic, what seeds would you seek out the most?
Oh, wow, that is a very deep question. It would have to be wild species. Crop species are not very resilient to harsh climates or to adverse conditions. I would use seeds from indigenous crops that people have been using for thousands of years, which are very stress resistant, which have a lower yield in general, but they would grow under almost all conditions. Okay.
Okay, yes, thank you for the response. Now, another question from Sulis Chandra. Professor Hilhorst, what are the most popular biological and chemical stimulants used in climate-smart agriculture?
Oh, now that is a question that only seed companies can answer. That is, most of these biologicals are secret compounds that seed companies have developed. And some of them are endophytic bacteria that can be used. But really, well, I'm not involved in that kind of research, but particularly at seed companies, this research is being done. And of course, when they find something that is beneficial to the seeds, they will not tell us. That is very difficult. Yeah, sorry. Okay, well, what's going on now?
Okay, we have a technical problem here.
Ah,
I'm sorry, Dr. Hai. Blackout.
now it's okay.
But we use sensor. Yes, now it's okay. Okay, now is the yes. We go to next question, Dr. Hai.
Yeah, sure.
Okay, sir. Okay, from M. Alif Kaisar.
I see it
have
here.
read some. Yes, Alif Kaisar. I have read some plant species do not develop after fertilization, such as the wild banana. What caused something like this, and what is the solution?
Yeah, that happens. Well, you have to find out if the fertilization is successful or not. Sometimes fertilization doesn't happen because of the temperature, the wrong temperature, or also the wrong morphology, particularly in species that are the result, crop species are the result of breeding. The morphology can be wrong or difficult. I cannot give you an answer here. This is not my field. But yes, very often lower yields of crops are the result of bad pollination. Yeah.
Okay, thank you, Dr. Yud. And now we go to another question from Natalia Uli Nadeak. I want to ask, how long does the seed usually have dormancy? Is there a difference between seeds that experience dormancy and seeds that fail or can be said to no longer grow?
That's also a very good question. How long seeds usually have dormancy, that may differ from 1 week to 10 years. It really depends on the species. As I have said, crop species, seeds from crop species usually have no or very little dormancy. That has been selected out during domestication. And then is there a difference between seeds that experience dormancy and seeds that fail? Now, if seeds are not dormant and they still don't grow, they must be dead or badly damaged inside. Yeah, that's my answer.
Okay, so is there any difference between seeds that experience dormancy and seeds that fail or can be said to no longer grow?
Yeah, no, that's what I'm saying. If the seeds are not dormant and they don't germinate, they must be dead or very badly damaged. Yeah.
Okay, next question from Handi Budiman.
Yeah.
I want to ask about how the mechanism of desiccation tolerance happen in plant.
That is also, I like that question because I'm working most of my time on this phenomenon of desiccation tolerance. And it is a very complex mechanism. We know a little bit about it now. For seeds, what we do know is that seeds can be dried because they have lots of protective compounds like sugars and all kinds of very specific proteins. But the most important thing is that in seeds, you have an accumulation of reserve food like starch and proteins in large vacuoles. And they are stored as solids. And that means that during drying, the cells of the seeds cannot shrink. They keep their integrity. And that is very likely the big secret for the seeds, but we're still working on it.
Can I ask another question, Mr. Hang?
Yeah.
So the desiccation tolerance is about the water is coming out of the seed from plants, is that right?
Yeah.
So how much does it will affect how much desiccation tolerance will affect the dormancy of the plants?
Oh, that is also a very good question. But what we think now is that desiccation tolerance and dormancy are very much related because both are mechanisms for seeds to survive adverse conditions. And when we look at the genes that are involved in desiccation tolerance and the genes that are involved in dormancy, they overlap. Many of those genes, at least 50–60% of them, are the same genes. So desiccation tolerance and dormancy are very closely related. Does that answer your question?
Yes, that's
Yes.
Thank you for the answer.
Okay, you're welcome.
Okay, thank you. Next question is from YouTube, from Tristi Indah. How to improve the seeds which can be adaptive in climate change? In the other way, the laboratory in our institution does support. How to improve the seeds which can be adaptive in climate change?
Yeah, that is the $1,000 question, of course, that I mentioned. We have to work on that, and we can use. Well, we have to understand the processes in seeds, how seeds can germinate under stress conditions, and also how crops can grow under stress conditions. We have to do the basic research, the fundamental research of all kinds of processes in the plants and in the seeds. When we know the genes, the key genes, we can start with setting up breeding programs, focusing at those genes or loci, or we do the gene editing the quicker way, focusing on those same genes. But first, and that's the important thing, we have to find the genes that determine whether a plant grows under high temperature or not. And the same for the seeds. Yeah.
Thank you, Dr. Hein. Now, the last question from Fabriliana. What if we use the early dormant seeds for cultivation of plant production? Is there any effect for the product result?
Sorry, can you repeat the question, please?
What if we use the early dormant seeds for cultivation of plant production? Is there any effect for the product result?
So if there is an effect of dormancy on plant production, I'm not sure if I understand.
For cultivation of plant production, what if we use the early dormant seeds for cultivation? Is there, what if we use the early dormant seeds for cultivation of plant production? Is there any effect for the product result?
Early dormancy, you say?
Early dormant seed, yes.
Early dormant seed. No, because what you very often see in developing seeds is that they develop dormancy. And then at the very end of the maturation period, when the seed becomes ripe, the dormancy disappears again. And as I said before, it's very difficult to use dormancy in your plant production programs because it is so unpredictable. So one year you may have very dormant seeds, and the next year from the same crop, the same cultivar, you may have seeds that have very little dormancy. So very difficult to say.
Okay. Okay. Thank you, Dr. Hank. It's time to end. Thank you, Dr. Hank, for the fruitful discussion
Okay, you're
and
welcome.
comprehensive response. And also thank you for the question from participants. Thank you, Dr. Hank. Have a nice day. Now,
Thank you very much. You too.
now we are going to the next agenda. The second presentation delivered by Mr. Danang Widyarso, SPMSC. Before beginning the presentation, I would like to inform briefly about Mr. Danang. Mr. Danang obtained bachelor and master degree from Gajah Mada University, Yogyakarta. His professional experiences are as variety trial researcher in 2002 up to 2004 at PT Benih Inti Subur Intani, and in 2004 up to 2011 as pepper plant breeder assistant at PT BC International Tbk, and now he is currently pepper plant breeder at PT BC International Tbk. So without any further ado, Mr. Danang will deliver his presentation for 40 minutes and followed by question and answer for 20 minutes. I would like to invite Mr. Danang. Time is yours.
Okay, thank you for your time. But in order to make the my explanation is clearly understand, I will use the bahasa Indonesia.
Yes, Mr. Danang.
Selamat sore Bapak Ibu dosen dan teman-teman sekalian. Berikut adalah presentasi yang akan coba saya sampaikan sehubungan dengan beberapa hal yang sifatnya praktis mungkin, sehubungan dengan yang tadi mungkin sudah disampaikan oleh Dr. Heng, yang ada hubungannya dengan perkembangan benih, lalu dormansi, dan kecambahan benih. Yang pertama, saya ingin mencoba untuk memulainya dengan menjelaskan bahwa di dalam bisnis pertanian biasanya ada beberapa departemen yang terdapat di dalam bisnis pertanian itu. Yang pertama adalah departemen riset, kemudian produksi benih, lalu quality control, processing atau pengolahan benih, dan marketing. Tetapi biasanya yang kalau kita bicara tentang benih, tiga departemen di atas itu yang lebih banyak bersinggungan. Sementara untuk departemen yang lain itu lebih banyak berbicara tentang varietas. Kompetisinya adalah kompetisi varietas. Sementara untuk departemen produksi, processing, dan quality control itu akan lebih banyak berhubungan dengan kualitas benih, berkompetisi dari sisi kualitas benihnya. Mungkin yang ingin saya jelaskan bahwa kalau kita bicara benih yang dijual untuk ditanam oleh petani, itu merupakan satu proses yang panjang, mulai dari menanam benih induk, kemudian melakukan polinasi atau perbanyakan budidaya tanaman itu sampai menghasilkan buah. Kemudian buah itu dipetik setelah dia matang dan kemudian diambil benihnya. Oleh karena itu, industri benih ini juga merupakan satu industri yang unik, oleh karena yang dihasilkan adalah benda hidup, sehingga mungkin tidak sama seperti industri yang lain yang kalau mereka mencoba untuk mencetak sesuatu menggunakan mesin, kemudian hanya memasukkan bahan-bahan tertentu dan itu sudah akan langsung jadi dan bisa terhitung dengan lebih presisi berapa yang akan dihasilkan. Sementara untuk perusahaan benih oleh karena prosesnya sangat tergantung oleh banyak hal. Tadi mungkin untuk benih ada banyak hal yang mempengaruhi, dan terutama faktor lingkungan itu menjadi satu penentu juga, sehingga ini menjadi satu hal yang lebih rumit. Secara umum kita akan melihat bahwa satu tanaman, kalau kita melakukan satu produksi benih, itu akan banyak dipengaruhi oleh faktor-faktor lingkungan yang ada di sekitarnya. Bisa berupa cahaya, kemudian air, dan nutrisi, kemudian oksigen, dan karbon dioksida juga. Dan kita melihat bahwa ada proses fotosintesis yang berpengaruh ke hasil benih yang diharapkan, yang nantinya juga akan berpengaruh di viabilitas benihnya. Kemudian ada respirasi dan transpirasi, ini yang akan menentukan juga kesehatan tanaman itu. Apabila laju respirasi dan laju transpirasi misalnya tinggi, sementara tanaman tidak mendapatkan suplai air yang cukup, dia juga akan mengalami masalah di dalam pembentukan benihnya, sehingga kualitas benih juga akan terpengaruh dan akan mengalami permasalahan dari sisi viabilitas. Ya, ini adalah satu gambaran, mungkin ada dari satu penelitian yang dilakukan tentang pengaruh dari panas atau heat stress terhadap pertumbuhan tanaman. Jadi oleh karena Indonesia ini adalah daerah tropis dengan intensitas penyinaran yang tinggi dan juga temperatur yang tinggi, kemudian produksi benih biasanya juga kami lakukan di dalam greenhouse, sehingga suhu rata-rata harian itu bisa sampai melebihi 40 derajat kadang-kadang pada musim-musim tertentu. Ini menyebabkan tekanan lingkungannya menjadi sangat tinggi di dalam greenhouse itu. Sementara untuk kita mencontohkan di sini untuk tanaman cabai, pertumbuhannya suhu optimum untuk pertumbuhan tanaman cabai yang optimal itu adalah sekitar 21 sampai 33 derajat. Dan suhu harian di atas 38 derajat ini akan menurunkan proses fisiologis termasuk proses fotosintesis dari tanaman itu. Nah, berikut adalah satu hasil penelitian yang menunjukkan bahwa memang heat stress atau cekaman panas, cekaman suhu tinggi, ini menyebabkan benih mengalami penurunan viabilitas yang sangat drastis. Jadi kita melihat bahwa untuk tanaman yang terpapar oleh suhu harian sekitar 38 derajat Celcius, mulai dari antesis, jadi mulai dari bunga mekar sampai waktu panen, ini akan menghasilkan berat buah yang paling rendah, kemudian pembentukan biji yang paling rendah, kemudian juga persentase benih abnormal yang paling tinggi, kemudian berat kering benih yang rendah, serta jumlah total karbohidrat yang paling rendah. Dan yang paling menarik adalah perkecambahannya ini sampai mencapai 0%. Artinya tidak ada satupun benih yang mampu tumbuh atau viabel oleh karena tanaman itu terpapar pada suhu tinggi di atas 33 derajat. Mulai dari bunga mekar, artinya mulai dia mengalami polinasi, sampai dia siap dipanen. Nah, ini adalah beberapa contoh untuk benih cabai yang oleh karena terekspos ke suhu yang tinggi akan mengalami permasalahan ya. Kita melihat Di sebelah kanan ini adalah benih yang abnormal, jadi tidak bernas, kemudian embrionya juga tidak sepertinya tidak berkembang dengan sempurna, dan yang pasti tidak viabel. Sementara yang di sebelah kiri adalah benih yang normal, yang bernas. Semakin tinggi tingkat stres atau cekaman panas yang dialami oleh tanaman, jumlah benih abnormalnya akan menjadi semakin banyak. Dan dari sisi produktivitas benih, akan merupakan satu kerugian oleh karena produktivitas benihnya akan menurun sangat tajam. Sehingga ya perlu dipikirkan tentang bagaimana cara untuk mengatasi permasalahan ini. Oleh karena memang secara alami Indonesia karena berada di daerah tropis, permasalahan tentang suhu tinggi atau cekaman suhu tinggi ini memang menjadi satu tantangan tersendiri untuk kita. Ini adalah beberapa hal yang mungkin bisa kita lakukan. Jadi ini adalah satu teknik yang sudah dikembangkan di Israel. Sebenarnya sudah mulai dilakukan mungkin sekitar awal tahun 2000-an. Kemudian ya karena situasinya mungkin sama di sana juga mungkin lebih panas dibanding Indonesia, ada risiko yang sama dengan cekaman suhu tinggi, sehingga kemudian coba untuk dilakukan pengurangan intensitas cahaya untuk mengurangi suhu di bawah naungan. Yang dilakukan adalah menggunakan naungan, tetapi memang berbeda dengan yang selama ini ada di Indonesia. Yang lebih banyak digunakan adalah shading net yang dia hanya mengurangi sekitar 35% dari intensitas penyinaran itu. Sementara, dan memang ini agak sulit ditemukan di Indonesia oleh karena sebagian besar shading net yang beredar di Indonesia itu memang lebih banyak dikhususkan untuk tanaman anggrek misalnya. Itu dia menggunakan shading net yang mengurangi sampai 65%. Jadi terlalu rapat dan juga pengaruhnya sebenarnya tidak terlalu baik untuk tanaman oleh karena cahaya terlalu banyak dikurangi sehingga proses fotosintesis juga mungkin akan berisiko berkurang juga, mengalami penurunan juga. Jadi screen atau shading net ini akan menyaring dan memantulkan radiasi, selain juga bisa berfungsi untuk mengurangi tekanan lingkungan akibat iklim yang tidak cukup baik, serta proteksi terhadap serangga hama. Ada beberapa warna yang bisa menjadi pilihan dan ini juga memiliki pengaruh yang berbeda terhadap tanaman. Spektrum warnanya berbeda, ini akan menyebabkan pengaruh yang berbeda terhadap pertumbuhan tanaman juga. Mungkin ini bisa jadi satu hal yang bisa kita pelajari sama-sama dan bisa kita gunakan, kita manfaatkan. Jadi kalau selama ini yang ada di Indonesia adalah shading net yang hitam, ini hanya untuk mengurangi cahaya saja, tapi ada pilihan-pilihan untuk net yang transparan, ini akan menyebarkan cahaya tanpa mengubah spektrumnya, dan warna net yang berwarna ini akan menyebarkan cahaya dan mengubah spektrum, sehingga akan memberikan pengaruh yang berbeda kepada tanaman. Jadi seperti untuk penyaringan cahaya dengan net yang berwarna ini akan memberikan pengaruh yang berbeda karena warna-warna tertentu akan diserap, kemudian ada juga yang akan dipantulkan. Beberapa bisa yang seperti warna mutiara atau pearl white ini, dia bisa menyebarkan atau meneruskan warna translucent yang tidak terpecah spektrum warnanya dan bisa berfungsi dengan baik untuk tanaman. Beberapa contoh untuk penggunaan shading net di pertanaman, beberapa pertanaman. Kemudian yang selanjutnya adalah pengaruh nutrisi. Jadi kalau kita tadi melihat bahwa ada pengaruh cekaman lingkungan terutama untuk suhu tinggi terhadap perkembangan benih, kita melihat di sini bahwa nutrisi itu juga memberikan pengaruh yang cukup signifikan untuk perkembangan benih, kualitas benih. Di sini kita melihat dari parameter laju berkecambahan untuk benih tomat. Jadi ada sekian dosis pupuk N, P, dan K yang diberikan, dan kita melihat bahwa ternyata untuk dosis tertentu justru yang bukan yang paling tinggi. Jadi untuk 60 kg per hektar N, 17 kg per hektar P, dan 50 kg per hektar K, ini yang bisa memberikan laju perkecambahan yang tertinggi. Jadi kita melihat bahwa ada pengaruh nutrisi di sini, dan itu artinya adalah bahwa untuk melakukan produksi benih, kita perlu juga memilih nutrisi yang sesuai dengan jumlah yang sesuai juga, yang akan bisa mendorong tanaman induk ini menghasilkan benih dengan kualitas yang tinggi. Di sini bisa dilihat dari tingkat perkecambahan dan viabilitasnya. Kita melihat di sini untuk benih tomat ya, lebih sensitif, jadi ada 3 kategori benih yang normal, kemudian abnormal, ini sama seperti yang benih cabai tadi, tidak bernas dan embrionya tidak tumbuh, tetapi ada juga benih yang ukurannya kecil. Masih bisa tumbuh, hanya oleh karena ini bernilai ekonomis, sehingga bagi petani rata-rata mereka akan lebih suka kalau benih itu memiliki ukuran yang lebih besar, oleh karena kepastian perkecambahannya akan lebih baik. Kemudian selain tadi kita sudah bicara tentang produksi benih, ini adalah proses selanjutnya setelah produksi, kemudian buah dipanen, kemudian diekstraksi, dikeringkan, kemudian ada coating dan packaging. Jadi proses-proses ini pun juga harus melalui satu serangkaian standar yang tertentu supaya hasilnya maksimal, dan benih yang tadi sudah misalnya dari proses produksinya sudah dilakukan dengan benar, tetapi apabila ada sesuatu yang salah yang dilakukan di dalam proses ini, itu juga akan berpengaruh. Seperti misalnya untuk pengeringan benih, misalnya suhu yang terlalu tinggi atau pengeringan yang terlalu lama, ini akan juga berpengaruh ke viabilitas benih. Ada beberapa contoh masalah yang muncul dan ini kemudian memunculkan satu standar yang harus diikuti. Misalnya untuk ekstraksi benih tomat, benih tomat ini tidak boleh langsung diekstrak. Jadi setelah panen tidak boleh langsung kemudian dibelah dan diambil bijinya, tetapi biasanya akan disimpan selama 1 malam, dan tidak kelihatan di sini, tetapi biasanya sebelum disimpan, dia setelah panen dimasukkan ke dalam karung seperti ini, kemudian akan diinjak-injak supaya dia hancur, kemudian difermentasi selama 1 malam, baru kemudian besoknya dikeluarkan dan mulai dicuci. Jadi ini tujuannya adalah untuk menghilangkan lendir yang ada biasanya mengelilingi benih itu, supaya setelah fermentasi ini akan lebih mudah untuk dihilangkan. Oleh karena kalau lendir ini tidak bersih, dicuci, kemudian mengering, ini akan membentuk semacam polimer yang menyelubungi benih tersebut, sehingga benih itu akan sulit untuk tumbuh. Jadi ini salah satu hal yang menyebabkan dormansi juga, sehingga kemudian dilakukan fermentasi seperti ini untuk memastikan bahwa pada saat pencucian benih, lendir itu akan betul-betul tercuci dengan bersih, sehingga benihnya tidak akan bermasalah. Lalu yang kedua masih hampir sama, jadi untuk benih timun ya. Benih timun ini juga dilakukan curing atau penyimpanan seperti ini, tapi bukan ya ada fermentasi juga, tetapi untuk yang curing ini fungsinya agak sedikit berbeda. Jadi ini untuk mendorong supaya terjadi penyerapan nutrisi dari buah, badan buah itu oleh benih, sehingga bisa meningkatkan kualitas benih itu, terutama untuk benih-benih yang mereka masih belum masak secara fisiologis. Jadi hal ini juga terjadi juga dilakukan di tanaman semangka. Tanaman semangka ini juga dilakukan curing sekitar 2-3 hari. Dan memang dengan perlakuan seperti ini, benih yang semula masih muda misalnya, pernah diamati, itu bisa mengalami perubahan. Jadi kemudian bisa benih itu menjadi masak begitu oleh karena curing ini. Sekalipun buah ini sudah dipanen. Kemudian yang faktor yang lain yang berpengaruh di viabilitas benih untuk timun adalah pengeringannya. Jadi setelah ini dicuring, kemudian dibelah, diambil benihnya, difermentasi, kemudian diekstrak, dicuci dan dikeringkan, dan setelah itu dijemur. Penjemuran ini juga harus melalui proses yang tepat. Biasanya ada yang melakukan penjemuran dengan langsung menggunakan alas di atas lantai jemur yang cukup panas, dan ini ternyata berpengaruh besar terhadap viabilitas benihnya. Oleh karena kemudian setelah pengeringan, benih ini akan menjadi mati biasanya oleh karena terkena suhu tinggi dari lantai jemur itu. Sehingga untuk pengeringan ini pun perlu dilakukan dengan benar. Ada 2 metode sebenarnya yang saya tampilkan di sini adalah pengeringan dengan menggunakan matahari. Ada juga yang menggunakan dryer, mesin pengering. Untuk yang matahari ini memang tidak boleh langsung bersentuhan dengan lantai jemur supaya tidak menyebabkan tingkat kematian benih yang tinggi. Kemudian yang lain ada juga permasalahan yang muncul misalnya di benih paria. Jadi untuk benih paria ini memang tidak biasanya hanya dilakukan curing tidak lama, tapi tidak dilakukan fermentasi. Biasanya panen dilakukan dengan membuka buah, kemudian mengambil benihnya, dan kemudian langsung dicuci. Nah, tetapi setelah pencucian ini ternyata tidak boleh langsung dijemur di bawah sinar matahari, misalnya. Oleh karena apabila setelah pencucian kemudian pengeringan itu dilakukan langsung, akan terjadi pengeringan yang cepat dan itu akan menyebabkan kulit benihnya itu menjadi lebih keras, lebih kering, lebih keras, kemudian sehingga sulit untuk menyerap air, tidak bisa terjadi imbibisi, dan ini menyebabkan benih itu dorman juga. Sehingga yang perlu dilakukan adalah setelah ini diambil kemudian dibersihkan, dicuci, perlu dilakukan proses kering angin dulu 1 malam dan setengah hari, kemudian baru dikeringkan di bawah matahari atau di dryer. Cara yang lain adalah bisa menggunakan alat seperti sentrifuge ini. Jadi sama seperti pengering untuk pakaian gitu ya di mesin cuci. Jadi cairan airnya dikurangi lebih dulu, baru kemudian dikeringkan sehingga tidak terjadi kehilangan air yang drastis yang menyebabkan pengerasan kulit biji. Yang lain misalnya untuk benih lettuce, ini juga karena benihnya kecil, biasanya kalau itu ditaruh di lantai jemur yang panas, ini juga akan menyebabkan dormansi dan perlu dikeringkan dengan cara tertentu. Nah, ada beberapa rekomendasi untuk pemecahan dormansi dari benih lettuce ini, yang menggunakan kombinasi CA3 dan sitokinin, dan ada juga yang mengatakan bahwa dengan penyinaran cahaya dengan diberikan cahaya ini akan bisa memecahkan dormansinya. Yang lain yang juga dilakukan salah satunya adalah dengan menggunakan dry heat treatment. Jadi ini adalah pemanasan benih yang dilakukan dengan menaikkan suhu secara bertahap, juga menurunkan suhu secara bertahap dalam waktu tertentu, biasanya bisa sekitar 2-3 hari. Metode ini sebenarnya awalnya didesain untuk mengeliminasi atau menghilangkan virus-virus yang seedborne atau yang virus-virus yang tular benih. Tapi kemudian diketemukan juga bahwa ada pengaruh perlakuan ini terhadap peningkatan laju perkecambahan benih. Artinya sayangnya memang untuk benih-benih yang mengalami atau yang sudah diberi perlakuan ini, dia akan mengalami pengurangan atau penurunan di daya simpannya, dan biasanya direkomendasikan untuk segera ditanam setelah diberi perlakuan ini. Lalu untuk pengujian viabilitas benih, kita masih menggunakan cara yang biasa dilakukan, saya pikir. Jadi dengan menggunakan tetrasolium test, ini dilakukan di Laboratorium QC. Ini untuk benih buncis, jadi yang bagian-bagian benih yang dia bisa mengabsorpsi warna merah ini adalah sel-sel yang viabel, sementara yang tidak mengabsorpsi warna adalah bagian sel yang tidak viabel. Dan kita akan melihat bahwa kalau bagian embrionya ini putih, berarti dia sudah tidak bisa berkecambah. Ini contoh untuk benih cabai yang diuji tetrasolium. Kita melihat di bagian tengah ini adalah embrionya, dan terlihat di sana untuk benih-benih yang dia tidak bisa menyerap warna merah ini adalah embrio yang tidak viabel. Kita masih menggunakan ini, masih tetap digunakan. Kemudian pengujian yang lain adalah dengan melakukan uji perkecambahan radikula. Ada yang dengan skarifikasi, ada yang tidak diskarifikasi, dan kemudian dibandingkan hasilnya. Jadi untuk benih semangka ini memang ada beberapa benih yang kemudian bisa menghasilkan benih yang keras hard seed, kulitnya kemudian menjadi keras. Dan biasanya kalau di petani itu praktek itu sudah berjalan, terutama untuk benih-benih yang triploid. Ini biasanya perlu dipecahkan kulitnya, bukan digosok ya, tetapi dipecahkan kulitnya sehingga ada celah yang nanti akan dipakai untuk proses imbibisinya. Kemudian ini adalah beberapa percobaan yang dilakukan untuk pemecahan dormansi juga. Ini adalah percobaan yang dilakukan untuk memecah dormansi benih bayam. Jadi diperlakukan dengan giberellin, dan terlihat ada bedanya. Perendamannya dilakukan selama 1 malam, dan terlihat bahwa ada pengaruh yang signifikan antara yang diperlakukan dan tidak diperlakukan. Jadi memang tadi seperti mungkin sudah dijelaskan oleh Dr. Heng bahwa aplikasi giberellin ini mungkin salah satu cara yang standar untuk melakukan pemecahan dormansi, gitu ya. Banyak diterapkan. Ya, berikut mungkin adalah kesimpulannya bahwa produksi benih ini adalah satu proses yang unik dan terperinci untuk memperoleh benih yang viabel, yang hidup, ya, dan juga seragam, bisa memastikan bahwa benih itu akan bisa menghasilkan tanaman yang sehat dan produktif. Nah, beberapa hal yang perlu diperhatikan, terutama di dalam produksi benih, adalah dari proses ekstraksi benih dan pengeringan benih yang harus dijaga betul-betul supaya benih yang dihasilkan ini memiliki kualitas benih seperti yang diharapkan. Demikian presentasi dari saya. Terima kasih. Saya kembalikan kepada moderator.
Sebentar, nggih, Pak.
Baik. Ya.
Okay, thank you, Mr. Tana. Now it's time for the question and answer from Bu Lita, Profesor Lita Stopo. Bagaimana prospek penggunaan shading net berwarna untuk produksi benih di Indonesia? Karena di Dubai sudah menggunakan variasi cahaya lampu pada vertical farming.
Baik, terima kasih untuk pertanyaannya, Ibu. Kalau saya pikir dari sisi prospek sebenarnya memang ini adalah satu teknik yang relatif tidak mahal, gitu. Hanya memang salah satu kendala yang kita hadapi sebenarnya lebih ke supply, gitu ya. Kami sudah mencoba untuk mencari shading net ini yang dengan warna tertentu dan kerapatan tertentu, tetapi memang kebanyakan supplier ini mereka hanya menyediakan tipe yang hanya bisa menyerap sekitar menyerap sampai 65% gitu. Kami sudah pernah mencoba impor dari Cina untuk shading net yang 35%, hanya waktu itu yang datang bukan yang berwarna seperti yang kami harapkan. Tetapi memang dari yang sudah kami coba dengan shading net yang berwarna hitam ini relatif bisa mengurangi tekanan panas di dalam greenhouse. Kami coba di dalam greenhouse. Hanya memang kalau dari apa yang saya baca dengan shading net yang berwarna itu sebenarnya ada lebih banyak keuntungan dibandingkan shading net yang berwarna hitam itu. Oleh karena kalau berwarna hitam dia hanya akan menyerap atau mengurangi intensitas cahaya saja, tapi tidak memberikan pengaruh positif yang lain. Sementara untuk tadi memang sudah ada teknologi yang menggunakan LED light misalnya. Tadi mungkin bukan hanya di Dubai, cukup banyak tempat yang sudah ada banyak negara yang menggunakan itu, dan saya pikir di Indonesia pun sudah mulai ada yang mencoba menggunakannya. Ada beberapa laboratorium misalnya, laboratorium kultur jaringan itu ada yang sudah menggunakan LED light ini. Tapi memang sementara ini kalau untuk LED light-nya, saya pikir lampu LED-nya itu masih bisa diperoleh gitu ya. Saya pernah mencoba melihatnya, tetapi memang untuk mencari variasi warna, misalnya untuk LED light ini, yang masih butuh penelitian lebih lanjut. Jadi kombinasi warna biru, warna merah misalnya, dan warna yang lain, warna kuning, itu masih perlu diteliti lebih lanjut. Dan mungkin aplikasinya untuk petani bisa jadi itu tidak mudah. Kalau untuk smart farming atau precision farming itu pasti bisa dilakukan. Tapi pasti hal itu akan membutuhkan biaya yang lebih tinggi. Nah, sementara shading net ini bisa memberikan pilihan yang lebih murah. Seandainya memang supply-nya ada supplier yang bisa menyediakan, ini akan menjadi pilihan yang lebih murah untuk petani dan tidak tergantung dengan suplai listrik dan sebagainya. Hanya memang ya ini menjadi pilihan dua-duanya antara LED light dan shading net ini. Kalau LED light memang saya pikir itu lebih banyak ke mengatur spektrum cahayanya yang diterima oleh tanaman sehingga bisa meningkatkan pertumbuhan tanaman. Tapi untuk yang shading net ini ada fungsi yang lain, yaitu untuk mengurangi suhu lingkungan yang ada di bawah naungan itu. Oleh karena ya ada permasalahan terbesar kita di daerah tropis adalah suhu yang tinggi. Kadang-kadang untuk beberapa petani yang dia sudah mulai menggunakan greenhouse misalnya, ini tekanan suhunya akan menjadi lebih besar oleh karena efek dari greenhouse effect itu memang menyebabkan suhu di dalam greenhouse itu menjadi jauh lebih panas, dan itu pengaruhnya memang cukup besar untuk tanaman. Memang greenhouse didesain biasanya petani yang menggunakan memang memaksudkan itu untuk mengurangi tekanan lingkungan yang lain seperti serangan hama dan penyakit. Tetapi kemudian ada efek samping juga berupa peningkatan suhu lingkungan. Demikian.
Oke, terima kasih atas jawabannya. Apakah Prof. Rita, apakah ada pertanyaan tambahan? Oh, sudah cukup. Terima kasih. Oke, pertanyaan berikutnya dari YouTube, dari Junior Lew. Izin bertanya, bagaimana sisa buah yang sudah diambil bijinya saja? Apakah ada produksi kembali dari sisa?
Untuk pengalaman yang terjadi pada kita biasanya untuk tanaman-tanaman tertentu seringkali memang itu masih bisa dimanfaatkan. Contoh misalnya seperti cabai, karena prosesnya adalah proses kering, ada yang kemudian mengumpulkan sisa kulit buahnya dan itu masih bisa dimanfaatkan. Beberapa seperti yang buah yang bisa dikonsumsi seperti melon, kemudian semangka, kadang-kadang ada yang melakukan itu juga. Jadi hanya diambil buah karena itu bijinya hanya terletak di bagian tengah, di plasenta, kemudian sisanya ada yang mengambil gitu. Tetapi ada jenis-jenis lain seperti yang kemudian dihancurkan, seperti tomat tadi, itu memang kemudian menjadi limbah. Satu solusi yang kemudian kita lakukan biasanya adalah dengan melakukan pengomposan, gitu ya. Jadi limbah ini kemudian masih bisa dilakukan pengomposan untuk mengurangi risiko pencemaran untuk perairan di sekitarnya. Kalau untuk beberapa jenis yang lain memang masih bisa dimanfaatkan. Ada seperti labu atau waluh misalnya, itu juga masih bisa dimanfaatkan sisa buahnya karena hanya diambil bijinya dan itu hanya terletak di bagian tengah. Demikian.
Oke, atas jawabannya. Enggak. Mati. Kalau langsung mati emang bisa ya? Enggak. Mati.
Oh, mati ya?
Sebentar, Pak.
Mohon maaf Pak Danang, ada mati lampu bentar tadi.
Baik, maaf saya pikir jaringan saya yang bermasalah.
Oke, ya apa ada pertanyaan lain? Silakan untuk bertanya secara langsung dengan raise hand atau bisa melalui chat untuk partisipannya. Yang di YouTube bisa bertanya melalui komen. Ya, partisipannya silakan bertanya. Bisa bertanya secara langsung dengan raise hand atau bisa melalui chat. Oke, dari Aditya Fadlani, izin bertanya ke Pak Danang, apakah sudah dilakukan upaya modifikasi genetik terhadap benih yang ada? Jika sudah, apa saja kendala yang dihadapi dalam upaya memodifikasi genetik benih? Terima kasih. Silakan
Ya,
langsung
baik.
direspon, Bapak.
Kalau untuk modifikasi genetik tidak. Artinya kalau kita bicara tentang genetic engineering atau rekayasa genetik, yang konteksnya nanti untuk meningkatkan kualitas benih, misalnya seperti yang tadi disampaikan oleh Dr. Heng, itu masih belum kami lakukan. Kalau dikatakan kendala, memang mungkin bukan masalah kendala, tetapi lebih ke kesiapan infrastruktur dan lain sebagainya untuk modifikasi genetik ini, oleh karena proses itu ya itu menjadi satu hal yang menjanjikan, tetapi juga butuh persiapan yang cukup banyak, terutama dari sisi peralatan, kesiapan dari mereka yang mengerjakan itu dan lain sebagainya. Selain trait atau sifat-sifat yang perlu dikembangkan atau yang perlu dimodifikasi begitu ya. Itu yang mungkin menjadi tantangan atau menjadi PR untuk dikerjakan. Kalau dari sisi kendala, saya pikir bukan masalah kendala, tetapi lebih ke kesiapan untuk bisa melakukan teknik tersebut. Ya, demikian.
Oke, terima kasih. Berikutnya pertanyaan dari atau Aditya Fadlani, apa ada tanggapan? Aditya Fadlani? Oke, terima kasih. Oke, berikutnya ke pertanyaan berikutnya Teuku Muhammad Farhan, dari Teuku Muhammad Farhan. Dari proses pengambilan benih tersebut, kemana limbah dari buah tersebut setelah diambil benihnya?
Ya, baik. Jadi mungkin sudah sempat saya terangkan. Jadi untuk limbahnya itu memang biasanya limbah-limbah yang memang tidak bisa dimanfaatkan itu akan kita komposkan gitu ya. Kita komposkan. Sementara untuk yang masih bisa dimanfaatkan seperti yang memang daging buahnya masih tersisa, seperti benih untuk pumpkin misalnya atau walu, kemudian kadang benih melon dan semangka, itu daging buahnya masih bisa dikonsumsi. Biasanya itu akan dibagikan ke tenaga kerja yang mengerjakan itu. Ya, demikian.
Oke, itu ada tanggapan? Oke, baik, terima kasih. Oke, sambil mungkin ada peserta yang ingin bertanya secara langsung kepada Bapak Danang, dipersilakan raise hand terlebih dahulu, atau juga bisa chat melalui Zoom. Ya, bagi peserta dipersilakan untuk bertanya secara langsung dengan raise hand terlebih dahulu, atau bisa disampaikan melalui chat atau komen kalau berada di YouTube. Oke, Bapak Danang, sepertinya waktu juga sudah habis. Sepertinya tadi pertanyaan terakhir dari Teguh Muhammad tadi.
Baik.
Oke, terima kasih Bapak Danang atas presentasinya dan atas respon jawabannya terhadap pertanyaan-pertanyaan dari partisipan. Sekali lagi terima kasih sudah meluangkan waktu untuk guest lecture hari ini.
Sama-sama.
Oke, sekarang waktunya untuk agenda terakhir, yaitu penutupan. Atas nama panitia, saya mohon maaf apabila ada technical problem selama guest lecture hari ini. Dan terima kasih telah menghadiri guest lecture sore hari ini. Sampai jumpa di lain waktu. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih, Pak.
Terima kasih Pak Danang.
Sama-sama Pak Wisnu. Terima kasih juga.
Terima kasih Pak Danang.
Ya, terima kasih Mas Wisnu.